Hi everyone, Brenda Gustin here, and I just wanted to show you a little bit about how to transfer cultures from one type of medium to another. We call this process subculturing. So you can see right here, I have two different cultures, two different broth tubes. This one's cloudy and that one's not. This one has bacteria growing in it already, so that's what I'm going to use to transfer to my other types of media. This broth here is sterile, meaning it's free of microorganisms. And so if I want to grow more of this, I can transfer some of this into the second broth tube, and that would be subculturing. Sometimes you want to transfer your culture to a slant, and slants are kind of handy because they are a solid medium, but typically we put them in a screw cap tube, so the culture, once the culture grows up, you can close the lid and put it in the refrigerator and they don't dry up too quickly. And we might also want to transfer or subculture onto an agar plate. And typically we would do that for doing streak plates or getting a lot of bacteria in a short amount of time. So I'm going to show you how to subculture this bacterial culture onto these other kinds of media. We're going to use what we call aseptic technique, which is clean technique, aseptic, not dirty. And I'm going to try to use good technique so I don't contaminate my cultures, so I don't add any more bacteria to them or any fungi to them that aren't already there. We're going to use a tool called an inoculating loop. And an inoculating loop, I can see if you can see that, it's a wire that has a little loop on the end, and these have been used since the golden age of microbiology for transferring cultures. So they're a very handy tool. They're called an inoculating loop, starting with an I. I'm going to kind of tilt this down a little bit, start my Bunsen burner, and I'm going to sterilize my inoculating loop by flaming it. Flaming means burning all the microbes off the surface of the wire, and there are no living cells that can survive being incinerated, being flamed or burned. I'm going to do 2 of them right here just to speed myself up so I don't have to wait for them to cool. Okay, so the first thing I want to do is take my culture that has bacteria in it and transfer it to a sterile broth. So I'm going to take my cooled inoculating loop. They take about 30 seconds to cool. And I'm a right-handed person, so I'm going to use my right pinky, take the lid off of my culture, and I'm going to flame it open, which means I'm just going to pass it through the flame 2 times. I'm going to dip my inoculating loop in there, and I'm going to flame it closed, pass the opening through the flame 2 times. I do that just in case there's some bacteria from my hand or dust on the opening of the tube. Now I'm going to get my sterile medium, my sterile broth. I'm going to open that lid with my pinky. I'm going to flame it open, and then I'm just going to transfer this culture into that broth. I'm going to flame it closed, close the lid, and now my culture has been inoculated. Now it doesn't look cloudy yet because it hasn't grown, but I'll put those in the incubator and they will grow overnight. Now I still have bacteria on my inoculating loop, so I'm going to sterilize that. Okay. I'm going to take that same culture and show you how to inoculate a slant. So I'll get my inoculating loop again. I'm going to flame my inoculum open, flame it closed. I'm going to open my slant, flame it open, and I'm going to start at the bottom of the slant, and I'm just going to do a zigzag pattern from the bottom to the top. Flame it closed. Put the lid on. We can tighten the lid all the way, but most of the organisms that we'll be using will, in fact, all of them like oxygen. So I'm going to turn it halfway open to make sure there's some air circulation. Always remember to flame your loop before you put it down because there's still bacteria on it. Finally, I can transfer my culture to a plate, and you would do that the same way. It depends on if you want to do a streak plate or just do a plate where you have some bacteria on it without doing a streak plate. But again, I want to take my sterile inoculating loop, flame my inoculum open, get a little bit on the loop, flame it closed, and I'm going to lift up the bottom of the plate and transfer some bacteria to that plate and close it. Always remember to flame your loop before you put it down. And that's some basics on transferring culture and using good aseptic technique. I guess I didn't point out the aseptic technique things that I was doing. A couple things that are like, don't hover over your plates. When things are open, hold them away from you. Keep them pointed away from you, because as I'm talking, I can make aerosols. I don't want to make the aerosol into my medium. Don't touch things that don't need to be, that you don't need to be touched. Every time you touch something, if I were to touch the inside of this agar plate, maybe I touch the inside of the lid, I'm going to transfer bacteria to that. So be aware of things like that. I hope that helps you understand a little bit more about transferring cultures and using aseptic technique.